Under the leadership of Donald Brown in the early 1980s, scientists at Carnegie's Department of Embryology recognized the importance of studying gene function in the context of cells and organisms. They developed a suite of exciting new scientific methods to interrogate the functions of various animal and plant genes, including the use of a naturally occurring mutation-generating phenomenon called transposable elements, or jumping genes, that were first discovered by Carnegie Nobel Laureate Barbara McClintock. Their innovative work forever changed human understanding of biology and set the course for the future of science and medicine. Today, 4 scientists who are a part of this revolutionary era, Allan Spradling, Gerald Rubin, Susan Wessler, and Jennifer Lippincott-Schwartz, will share insight into the process and impact of the groundbreaking genetic tool development in the 80s, how their experiences at Carnegie shaped their illustrious careers, and why model organisms like the fruit fly are critical to understanding shared biology among animals, including humans.
Well, hello, everybody, and welcome. I'm Frank Cesno, and it is my great pleasure and privilege to be here today to be speaking with you, our wonderful audience, with our wonderful panelists about the wonderful Don Brown. As Yishan said, this is the second in a series of conversations and symposia to talk about his incredible contributions and where we are. I'd love to bring the panelists in now and introduce them to you. Let me start with Dr. Susan Wessler. Sue Wessler is the Distinguished Professor of Genetics at the University of California in Riverside. Hello, Sue. Are you coming to us from California?
I am coming to you from California, rainy California today.
Well, and rainy California, that's actually a good news story for a change, isn't it?
Very good news.
I hope you get a lot of rain, and I hope California gets a lot of rain. Looking forward to the conversation here. Up next, Dr. Allan Spradling. He's Director Emeritus of Carnegie Embryology, where he's a principal investigator. And Allan, welcome to you.
Welcome.
And you are coming to us from where?
From Baltimore.
Which is a good place, not completely coincidental here, correct?
Not if you're talking about the Department of Embryology and Don Brown.
Okay, well, more to come on that. And finally, joining us is Dr. Gerald Rubin. He's a senior group leader at Howard Hughes Medical Institute, Genelio Research Campus, and got a lot of claim on jumping genes. So, Jerry, if you're there and you can unmute your camera, we'll be able to see you and greet you to the group. Let's see if he joins us. There we go.
Okay, here I am. So I'm talking to you from Leesburg, Virginia, where I now live and work at the Genelio Research Campus of the Howard Hughes Medical Institute.
That's great. That's great. Well, thanks to all of you. So what we'll do here is we'll talk a little bit about Don Brown and the lab and his impact on science, your science, and the remarkable work you all have done. We're also going to hear from Jennifer Lippincott-Schwartz. She's not able to join us here in person, but she's got a very interesting and informative video that we'll share in just a few minutes when we get down to that. I'd like to ask all of you, sort of, since we're recognizing the extraordinary role, contributions, influence that Don has had in many of our audience, I think, know Don, certainly know of his work. And I hope he's with us in the audience today. I kind of want to ask you what each of you think is the sort of the first thing the audience should know from your perspective about Don. And Sue, I'd start with you on that.
Well, I think Don sort of set the tone for having the lab leaders work in the labs. And I guess when I was leaving Carnegie, I was a postdoc there from 1980 to 1982. And on my last day there, Don came in and he said, I just have one piece of advice. He said, don't go on a hiring spree when you start your lab. He said, always remember that you're going to be likely to be the best scientist that you can have in your lab for many years. And so I really took that to heart and hired a technician immediately. And I worked with her for my first, 6 years, and that basically got me the papers for tenure. But I've made sure to tell young scientists that advice because I think it's extremely valuable.
I mean, it's really interesting advice, and it sounds like you took it, right? His message to you was beware spending your time managing people, do the science.
Yeah, especially initially, because when you're new, nobody really knows of your lab, and you're not going to get the top people, and you will be that top person for many years. Yeah.
Allan, how about you? What's your Don Brown reflection?
Well, I've been at Carnegie long enough. I have a large number of Don Brown reflections, but let me just mention a couple related to when I first came to Carnegie. And it follows along with some of the things Sue was saying. Don really felt like that we should just be doing science. And I've never seen anyone who could organize an institute with a smaller amount of administrative distractions. We sometimes went for months without a faculty meeting and nothing bad happened. So just the sheer efficiency of doing everything else so we could just be in our labs working made a big impression. And another thing that made an impression, which is a little related to what we just heard, is Don was pretty frugal with money, but he would never balk at the idea of having anyone you were going to consider for your lab fly in and meet everyone else and give a talk, because he knew that these were some of the most crucial decisions that young faculty members made. And having an unfortunate person in your lab can really change the atmosphere and be a problem. So being as careful in selection as possible is a very high priority.
Jerry, I know the money bit resonates with you with respect to Don.
Yes. When I got to Carnegie, it was my second job. I started off at Harvard Medical School, and which was what you could do was determined by what you could raise grant money for. So when I got to Carnegie, Don sat me down in his office and said, you should think about what the most important science that you can do. You should not do what everyone else is doing. Determine for yourself what's important, and don't worry about money. I'll find the money for you. So this kind of faith in a young scientist, and to say, I'm here to help you, this is very unusual, is unusual. And I think what made Carnegie the way it was, and why so many people at Carnegie went off and did unconventional and pioneering research.
We are incredibly fortunate to have the four of you here today, and I'm sure our audience knows all of you well, but I want to double back a little bit on what each of you have done. And Sue, I want to start with you. You have done such incredible things, including cloning the first gene in plants, and your lab has broken ground in the sort of computational analysis of jumping genes. And I want you to talk a little bit about this and how you launched, because your launch, which was back in those days as a postdoc, you were inspired by the famous Barbara McClintock.
Yeah. So this was 1980, and she won a Nobel Prize in 83. So her ideas were really starting to permeate all areas of science. And when I got there, I entered Nina Fedorov's lab, and she had, through a couple of meetings with Barbara, decided to change the focus to start her new lab around the isolation of the maize transposable elements. At my first year there, Barbara visited for about a week, and it was just, it was just a real privilege. And
did you know that you were in the presence of a giant when she was there?
Oh yeah. Yeah. I mean, every time I would go see her, or anytime I saw her, I said to myself, remember this, because this is something that you're going to tell your children and grandchildren eventually. So no, I mean, she was, one of the 3 M's, Mendel, Morgan, and McClintock, really. So
how did she and her work, her research influence you and your trajectory in those?
It was a direct, Nina Fedorov had, as I said, focused her lab on isolating at the molecular level the mutant alleles that Barbara had isolated in the decades before. And so we really had to go in and I wasn't the first one to clone a plant gene. There were a couple before, but we had to clone a gene in order to isolate the elements that then sat in it. And that was very difficult. Everything back then was very difficult. This is pre-PCR. This is, Southern blots and making huge libraries and, no computational analysis, certainly not in molecular biology back then. So everything took, dissertations that you did then could be done in a day or two now. So yeah, it doesn't sound very impressive what we did, but it took a couple of years.
It was, yeah. And Allan, Jerry, what was it like working in those labs at Carnegie at those? I mean, this is before cell phones and email and social media and all the rest. These were different times.
Well, they were scientifically exciting times, but some of these limitations that Sue mentioned really slowed things down. Particularly in the case of the fruit fly, you could not put genes back into the animal and see what happened. And this is a crucial technical ability to do a lot of genetic approaches. So I would say the lack of the ability to transform, as they say, or to put genes back in Drosophila was the key limitation. The genetics was great, but not if you can't combine it with the molecular side of the science.
And Jerry, if you grab a glass of water, I want to make sure.
Sorry, I'm fine.
That's all right.
My allergies are really bad. So yeah, this really ties into Bob McClintock, because when I had come to Carnegie, I had an interest in transposable elements. And early on in my Carnegie time, we cloned the transposable element family, the P elements, which was very active. And we had a feeling we can control its activity. So by analogy to Bob McClintock's work, we thought we should be able to use this as a way of introducing genes into Drosophila by injecting, rather than having a mating between flies and the transposable elements move, we would engineer the transposable element in the test tube, which I like doing. And then Allan, who had great skill and patience, would inject these DNA constructs into the embryos, and we would hope that the transposable element would jump off our injected DNA into the germline chromosomes of the fruit fly and thereby be passed on to future generations. So this was the exciting experiments that Allan and I were doing at Carnegie, and it was, as Sue mentioned, Don encouraged us to work in the lab. So Allan and I were doing these experiments ourselves, and we considered it to be very risky science and not a good thing for young postdocs or students to do. So we said, we can afford to take this chance. We'll do this. And Don provided us the equipment. We didn't have to write a grant to do it. We didn't have to convince anyone else. All we had to do was convince Don, and he enabled us to do these experiments. And this was 40 years ago. And without doubt, for me in my career, this was the most significant science that I've had the opportunity to do.
Why was it so risky? Why was it not considered a good thing for young scientists to do?
Because many people had tried to make genetically engineered animals, and no one had succeeded. And it wasn't really clear why or how to do this, but we had this in from the transposable elements. Maybe Allan has a...
Well, they'd even tried transposable elements before, but you need to have the right transposable element. We now understand exactly what determines that or not, but there'd been false claims of success. We felt that we were going to do this, and we were not going to reveal much until we had absolutely incontrovertible evidence that we had been successful.
And that's another thing that Don did. I mean, as you had your aha moment, right? He encouraged you to collaborate and to share this publicly.
Well, yeah. I think there are a few things where the normal rules just don't apply. I mean, we had found something that, like I mentioned, it was holding back all of our work, and we knew this was true of everyone in the field. So it was hard to justify not making this available as quickly as possible, because the number of people who could suddenly be empowered to do much better science was large. And so how often in a career do you have a chance to suddenly make such an incremental push of science forward? We just felt we had to share it. Even it turned out we did share it, and it was 6 months prior to publication.
Wow.
But I think here Don Brown played a big role as well, because we were young kids. We were like 32, 30, whatever. And so we had a long career. And I remember a conversation where we sat down with Don and said, should we share this right away? Or, as it was then the norm in science, you waited till you publish something and then you shared it. And maybe we could have said, oh, in the 6 months it will take to publication, we could do all these nifty experiments. But Don sort of said, you know, I think it would be much better for you to share everything right away. And for me, at least, you know, that made a big influence that we were doing the right thing. And it turned out exactly to be the right thing to do.
It was a big. So did you know something was up? Something was going on with these guys before they shared all this?
Absolutely. I mean, we knew there was something secretive going on, and we kept seeing them going into the cold room and together and all of this. And then we had these weekly lab meetings. The whole Carnegie it was called nukes, the nucleus or nuclear meetings. And they were on Thursdays. And there was a sign put up and said that Jerry and Allan would be presenting, and that afterward there would be a party. And so we knew it was something very, very good. I remember it like it was yesterday. I mean, the room was packed. It was sort of the library eating area at Carnegie. The room was packed. They got up and made this presentation that just blew the doors off the place. I mean, it was incredible. And we all got up and gave them a standing ovation.
And what did you hear that was so incredible for people in this audience who weren't there?
Everything. They started from the beginning and went through from beginning to end, an application. It was something that you don't see in any kind of seminar, I think, where everything was new and risky and successful.
And did you gentlemen know how big this was? Were you sharing that, blowing the doors off? Allan?
Well, we knew it was the most important thing that either one of us had ever done. That was for sure. And I think it really, when other people, you know, were able to reproduce it and it started to permeate the field, then we had a much better sense. So the reason we're being secretive, we weren't paranoid. It's just that we wanted a little space because we felt this was risky, and there've been false claims of people having success and then went away. And we said, if people heard, we thought we had done it and then it wasn't really true, it would be bad for our reputations. So we really wanted to be sure that we had it right. We had done all the controls, we repeated it. We're 100% certain that what we're saying was true, which is unusual. As Sue mentioned, usually people give more progress reports when things are less complete. So we just held back until it was really done and then let it all out at one time. So it was an unusual way of doing it.
Allan, anything to add to that?
Well, yes. I mean, I think we didn't know, you know, what was going to come from having this ability. We didn't really know how genomes were organized in a way that now this technique could tell you. And over the next few years, the technology just proved to be even beyond what we could have imagined in terms of what it opened up. We particularly enjoyed the way that we gave it out, however, you know, because right when we had this conversation with Don and we all agreed we should give it away immediately, it just happened the Drosophila meeting was coming up, the national meeting where representatives of just many of the fly researchers would be present. So we made these little packs that had everything, instructions and strains, everything you needed to do transformation was in these packs. And then after we gave our similar presentation to the meeting like that we had at Carnegie, Jerry just announced that, oh, and by the way, you can pick up your transformation kit on the way out as you leave this talk. And people just were amazed. And then they all picked up their kits. And the next month, we got letter after letter after letter from labs. All that they thanked us for our generosity. They said it worked the first time, just like you said it would, which I think about sometimes when I hear discussions of whether biology is a reproducible science. And most importantly, they just went on and on about how this had changed their whole goals of their research program, and they just felt, they made tremendous step forward.
Wish I'd been there. It was just when you were doing this, just when CNN went on the air, because I work for CNN. We could have had live cameras there. Imagine how exciting that would have been. We're going to go to cameras right now. Jennifer Lippincott, Dr. Jennifer Lippincott-Schwartz, as I mentioned, couldn't be here, but she also was in so many ways launched by Carnegie and Don Brown. She's the senior group leader at Howard Hughes Medical Institute's Janelia Research Campus, founding member of the neuronal cell biology program at Janelia. So for a few minutes, let's listen to her comments. And I want to remind the audience that if you've got questions for our panel about their science, about Don, about how this science is done, about where it's all going, please use your chat function, your Q&A function. But for now, I'd like you to listen to Dr. Jennifer Lippincott-Schwartz.
Hi, everyone. I'm Jennifer Lippincott-Schwartz, and I'm delighted to be part of this celebration of the Carnegie Institute and Don Brown. I spent many years at the Carnegie as a graduate student, and it's been a very important part of my career. I'm currently now at Janelia Research Campus, where I'm a senior group leader, and I'm studying cell biology and the way, in particular, how subcellular organelles are coordinating various processes within cells. This topic actually is something that I've done my whole career, and Carnegie was a very special, had a really big influence in me in terms of first becoming really excited about subcellular organelles when I was a graduate student with Doug Fambrough there in the 80s. It was an absolutely amazing, amazing time. I only really appreciated that as I sort of progressed in my career and really realized what an incredible and powerhouse environment Carnegie was. In fact, I always think of Carnegie as the land of the giants. Part of it was I was a graduate student, but part of it was the people around me were just exhibiting every characteristic that you could imagine for what a great scientist is all about. Don Brown had, he allowed graduate students to actually organize a big seminar series. And I did that once. And it was like an incredible experience for me because I was able to invite Nobel Prize winners, like George Pilati, Jim Rothman, Gunther Blobel, to be speakers at the seminar series that I was organizing with another postdoc. And that gave me the opportunity to directly talk to these incredible scientists in the field of membrane trafficking, which was what I was interested in. And basically it really demonstrated, I thought, a real incredible sort of leadership at the top to allow younger people to do this sort of thing. So it was hugely impactful. I mean, frequently what happens I see at these bigger institutes is that students really close off. They only become completely confined to their own little world of science. But at the Carnegie, because we're walking around this relatively small campus environment, you're bumping into people left and right. And Jerry Rubin was sort of down the other hall. And what he was up to with Allan Spradling was really exciting. And that was like one of the highlights of my experience there was their sort of unveiling transposable key elements in a presentation to the whole institute before any publication or public announcement. We were sort of all brought into the auditorium and sat down, and they sort of laid out the work that they had been doing in the cold room for several months, and I had noticed that. I knew something was exciting because those guys were working around the clock, and graduate students notice when PIs are working around the clock in a cold room. Something must be going on pretty cool in there. And so it was really fun to see that and to realize, wow, this is a big achievement. I'm witnessing a major breakthrough. And so that was really, really exciting to be able to experience. And it was experiencing firsthand. Even though it was completely outside my field, I felt like I was part of it because I was part of the Carnegie environment that was really special.
A Carnegie environment that was really special. Let me ask our panelists to join us back, and thanks for those comments. And as we get Sue and Jerry and Allan back, as I'm listening to her and listening to all you, I guess the next question, well, the first question is, were you aware that the graduate students were watching you that closely? Because apparently you were under the microscope.
I wasn't aware of that, no. I just thought I was going into the cold room, and it's not really that pleasant in here, but this is the place to get the job done. And
the serious question I have for you is, what then was the reaction from other scientists? Like I said, we didn't have social media, we didn't have an internet. What was the reaction? How quick was the reaction after you did this?
That's what really surprised me, because as you mentioned, there was no internet, there was no way people would know, but there were telephones. And so I got phone calls. We did a lunchtime talk, 4 o'clock in the afternoon, I was getting phone calls. I heard this rumor, is it true, from California, friends of mine, other people. So it really spread for those days. News traveled pretty quickly. And so it was exciting, and we were overwhelmed. There was a lot of interest from the press. I remember there was a television crew that wanted to come, but we refused to let them come because we thought this is going to be a big distraction to have a TV news crew from one of the big networks come in. So we said, no, we're not interested in that. But there was a lot of various coverage in newspapers, some of it more accurate than others. And it was, we had the feeling that we had done something that not many scientists have the ability to do in their careers. I mean, there are many, many fine scientists who go on and do great work, but this was something when we knew we had done something unusual. And as I said, I've been a scientist running a lab for 40 years since, and this is still the peak moment of my scientific career.
Allan, what was your reaction to what was going on around you?
Well, I knew that we had done something really unusual, and that it was a combination of, taking a risk, but also being lucky, and especially that we were in the right environment to do this kind of work. And I don't think it would have happened in many places. I kind of, feel like it's almost like gardening. A lot of plants, you put them in your yard somewhere and they do okay, but then if you move them to that special place, all of a sudden they're like growing much faster, they're prettier blooms. And I feel like the Don's department was like that special place. And that, just the atmosphere and the encouragement and just the shared belief that anything was possible in this department played a factor in why we were able to accomplish that.
So Sue, Allan and Jerry are working with this remarkable stuff and making these breakthroughs. They're working with fruit flies. You're working with plants. What's the connection? How did this affect your work with transposons and what you were doing from there?
Well, we were working on the material where transposable elements were originally discovered. And we were working, my project and the project of another postdoc were to isolate the AC and DS elements of maize. And essentially, the entire time we were there was Jerry overlapped. I came about when Jerry came in 1980, and when I left my faculty position, he left also to go to Berkeley, which is unusual for a postdoc and a faculty member to have the same term. But when he got there, they started their lab meetings talking about how they want to isolate this element, and then they isolated it, and then they put it into practice. And in that time, we were struggling. I mean, we're working with a genome that was the size of the human genome with none of the advantages that you have now. And so, by the time I left, with 2 years, there were 2 postdocs, me and another one, we had managed to isolate part of the gene that, in all that time. So things just went slower with plants. But that work has sort of fueled my last 35 years running my lab.
Which we'll talk about, but on transposable elements, flies and plants, I guess I'd ask this to the three of you. How did you and your research inform the other? Was it in tandem? Were you building off each other? I mean, there you are together, right? So talk about that.
It's essentially the same story. I mean, transposable elements, whether they're in Drosophila or in maize or in worms, are essentially the same creatures biochemically. And so we were speaking the same language. The genetics was the similar or the same. It was, these elements have moved through the tree of life, and they unite, all organisms basically, and they are the majority of the genome of most organisms.
And we were all knowledgeable about Bob McClintock's work. So she was a common unifying language. We all knew what she had done. It guided the way we thought about our results and how we could use them to do genetic engineering. It was really the intellectual framework was all from a common source, which I think is another thing that brought us together.
Allan?
Yeah, and it's so many of the things that had been discovered by McClintock and other maize geneticists about transposons, we fairly quickly, once we had the P element system working, could easily investigate. And again, it turned out that it was just like maize, like things like a local jumping and, excisions that were imprecise and things like that. So this was very, very useful, practical previous knowledge that we could put together for the engineering of the Drosophila genome, in novel ways, but built on solid genetics from past work.
So, you've made this big splash, and you shared with me an article that was written at the time. This is from October of 1982, and it says, it's very ominous headlines here. The cloning of human beings is one step closer. This is coming off of your work. And then there's this editorial at about the same time, cloning must stop. It says, when Wilbur and Orville Wright announced their most famous achievement, some contemporaries offered the opinion if God had meant us to fly, if God had meant us to fly, he would have given us wings. Until recently, anyone who expressed a doubt about the benefits of science was treated with the same derision as those simple souls. And then it says, the prospect, which is new, opening before us, is too awesome for the state not to interfere. Until society as a whole has determined the conditions under which cloning is acceptable, if it ever could be, we shall be neglectful of the future of humanity if we allow these experiments to continue. Did you realize what you had stumbled into, and how did you respond to this?
Well, I think we all probably experienced similar situations. I remember somebody calling us up and saying, tell us about how you turned the color of fruit fly eyes from brown to blue. And of course, fruit flies don't have blue eyes. Humans have blue eyes. They have red eyes, so we turn them from brown to red. You know, I got an award in '82 from the Maryland Science Society, and I got the second. They give two awards, you know, the highest award and then the second award. So I went down to the ceremony, and the chairman of the panel privately whispered to me, most of us wanted you to get the biggest award, but one person on the panel said this is going to be such a disaster for society that he wouldn't vote for giving you the top award. I mean, there was just an undercurrent that that genetics is really dangerous, and people are not that happy to hear about advances always.
Jerry, and there's a question from someone in the audience who asks, you know, what was the really big significance of this and this genetic work that you had done? What was the breakthrough?
Well, in order to do certain kinds of experiments, suppose I isolate a gene and I want to prove it's the gene I think. The best way to do that would be take a mutant organism where that gene was inactive and introduce my clone gene in the test tube back in that animal and basically cure its genetic defect. So that's what the technology we developed allowed people to do. So one, it allowed us to confirm when we had isolated something was what we wanted, but then it also allowed us to ask questions about what happens if we express it in the wrong cells or express it at a higher level, or what will the effect on development be? So it allowed us to test the models where people were coming out with about how animals developed and by manipulating them in a sensible way. So I think it just opened up the experimental repertoire that was available to biologists that they didn't have with any real organism other than single-celled organisms like bacteria and yeast prior to that.
And Allan, the fruit fly was a critical player in all this, and a lot of scientists, as you mentioned, who were using the fruit fly were able to follow this work. What did that lead to?
Well, I would say if you can imagine a map of the genome at that time, you would just be looking mostly at white space. Because there had never been an assay that could correct a genetic defect, there are many aspects of animal genes that don't really have any analogs in the single-celled animals. Like, where's the regulation to tell you what cell type to turn the gene on in? What about the timing in a whole long developmental and complex life cycle? What about spatial patterns? Is there regulatory information for that? And suddenly, with our method, you could find where the regulatory information in the genome is. People used to always joke about how we only know what 2% of the Drosophila of the human genome does, because it's only 2% that actually codes for protein. Much more of it is regulatory information that answers those questions about what cell type should the gene turn on in. So we not only began to map those, but the amazing thing is they turn out to be very flexible, elements called enhancers. And once you know about them, you can use the same enhancer for other genes. You can port whole patterns of regulation to, say, a marker gene that makes so-called reporter gene. And so the first reporter genes were made as a result of this work.
Could you explain that? What does that mean?
It basically is a way that you use a very, very easy to visualize gene expression. Now it's often GFP, this fluorescent protein. And then if you want to see what an enhancer can do, you make it drive a GFP. And now in one very simple experiment, you can get a vast amount of information on the regulation of the gene or of what that enhancer can do. And all these things, the fact that genes would have multiple enhancers that separately drove one part of their program, but then another enhancer drove another part of their program, none of that was really known. And this tells you a lot about evolution. because it's much easier to imagine how a gene could acquire a new function by just bringing in a new enhancer, perhaps even on a transposable element. Then the current view at the time is you'd had to have one point mutation after another, and even then it was questionable whether that wouldn't destroy other aspects of its function. So that the modularity of genes was not appreciated before. And it was one thing like that after another, that once you had an assay, the picture of animal genomes just started to fill in.
So I think for me, the exciting thing is there are a lot of people working very hard to solve their problem of gene regulation or development. And we provided them with a technique, an extremely powerful technique to accelerate their work. So this is what coming up with a new technique, why I like that kind of science, because if you discover a fact, that's fine. There are a lot of facts and some facts are more important than others, but you discovered that fact. If you make a new technique that enables all the labs in your field to become more productive, it's a different kind of impact it has, and you make a lot of friends that way. So I think Allan and I, because of the way we just shared things, we became very popular in the scientific community, which was good for our careers, I think.
Well, I want to come and talk about your careers and some questions we're getting from the audience in just a minute. But Sue, before I do that, I want to ask you, since you were working on jumping genes and you were working in the plant realm, how this propelled your work, and what for you, how were you able to take this and translate this into breakthroughs in your research, in your science?
Well, I think one of the things that we had sort of discussed before was the idea of taking risks. And I think at that time, working on plant genomes was a bit of a risk because it was considered sort of the Wild West. And when you're kind of in the Wild West, everything is open to you then, just as Jerry and Allan are talking about, with this technique, they opened all these areas. Well, once we had access to the transposable elements, some of the genes, there were two things that opened up. One was all of the mutant alleles that had been isolated by McClintock and others became available. But then at about that time and a little later, genomics, people were starting to sequence much more than just genes, but whole genomes. And what we were finding was that, these transposons that were inserting into genes causing mutations were the minority, that in fact, the vast majority of the genome was in fact derived from transposable elements. And that's the question that has driven me for my entire career over the last 35, 40 years is, how is that possible? How can these elements amplify to the point where they're close to 50% of the human genome, they're 80 to 90% of some plant genomes. How does this happen and you still have a viable, healthy organism? So, how can an element expand like this to hundreds, thousands, tens of thousands of copies and still be compatible with life? So there's huge questions that were opened up by that. And now these are questions now that we can sequence whole genomes easily. And what I mean is not little pieces, but actually the PacBio techniques where we can look at members of populations. We see that transposons are involved in restructuring the genomes of individuals, of members in populations. And that it's beginning to be seen that the changes that they are responsible for are the ones underlying adaptation and sort of evolution in species and organisms.
Well,
So
and in the plant realm, this is vital as we're thinking, and we'll maybe get to this in a few minutes, thinking about climate change and thinking about the adaptation that we may be able to help accelerate through some of this. Let me go to some questions from the audience now, because we've got a few, and I'll encourage the audience again, if you've got a question, put it in the Q&A. This actually plays off of perhaps the article that I was throwing a moment ago. Kathy asks, were the lead scientists of in this presentation concerned at all about ethical issues such as eugenics?
Well, we were working on fruit flies, so we weren't really concerned about what our experiments in terms of studying fruit flies, I would say.
Or
Now,
where that could lead?
well, I mean, we were not, I mean, we would have been concerned. I mean, we never would have suggested doing this on humans, right? And so I think there was a general recognition among us, and I think everyone in the scientific community, that one shouldn't be doing this kind of genetic engineering on humans at that point. So I think that was like a generally accepted.
I think the reason you wouldn't want to do it on humans is a little different than what the general public thinks. I mean, the general public acts like the only thing that's kept us from not having a race of supermen is just not having a technique to manipulate genes. Whereas the reality is you could have the best technique you could possibly imagine for manipulating genes, and we have absolutely no idea how to make any human better in any way, except unless they have a mutation in a gene, then we can fix that. So the real reason you don't want to do it in humans is these techniques are not without some occasional collateral damage, and you're just going to cause a problem. You're not going to do anything good. So there's just no need for it, except in very limited circumstances, which, usual current medical, procedures for approving things could probably deal with, but it's just not going to be a big change in any way in human characteristics or society. It's just, unfortunately, biology is so much more complicated than people think. It's just not something we're even close to engineering.
Sue, anything you want to add to that?
Well, I was going to say, I mean, it's prohibited. It has been done. I mean, it has been done by Chinese scientists, and that person, I think he's in jail now. So it's prohibited. Yeah.
Okay. Here's another question. This is from Diane. A few years later, after you did your thing, Andy Fire brought his own molecular biology kits to the C. elegans meeting. Was this inspired by your sharing?
I don't know. You'd have to ask Andy. I didn't know that he'd done that, but it would have been in keeping from the Carnegie tradition. He certainly would have been aware of what we had done and took the lesson that this was the way to make friends and influence people.
Allan?
I didn't know either, but I'd just say, great. That's I'm glad if we did inspire something that positive and beneficial, that's wonderful to know.
DJ asked this question to both of you. How did your collaboration start? And in a different scenario, could such collaboration work if you were at a different institution?
So I might say it would have been difficult if we're not at the same institution, especially if we're at institutions that were unlike Carnegie. We each had our own grants and we're very worried about our own program and whatever. At Carnegie, we had the time. University faculty often have many other demands at the time. We didn't have time as to start working around, the clock on something together. And this came from our informal discussions at Carnegie. Allan and I came from very different mindsets and backgrounds. I was a pure molecular biologist and Allan knew developmental biology. And to get these experiments to work, we needed both Allan's abilities and my abilities together. No one person could have done this full range of its experiments. And we were there and we could decide to do it. We could go to see Dawn and say, Dawn, can we take over this cold room? Will you give us this money to buy this piece of equipment we need for injecting the embryos? Fruit fly eggs are very small. You have to inject them under a microscope with a fine needle. So it was really a coming together that I'm not sure how easily it would have been, elsewhere. If we had been assistant professors at universities, I don't think we could have done it, but maybe Allan.
Well, I agree. It taught me that to have a great collaboration, you need someone who you're compatible with personally and you can absolutely trust, which I felt both of us had that feeling. And then you need people with different skills, because otherwise you haven't really incremented your total capabilities that much. And this, I've never experienced a collaboration as good as this was with Jerry, but I would try to follow these principles if I was, you know, in the future when I'm looking for collaborators, to follow those two principles.
Sure. Again, a reminder to the audience, if you've got a question about jumping genes or about the genome or any of these things that we've been talking about, please use the Q&A. We've got both our fruit fly conversation going here and plant. And Sue, I want to come to you on the plant genetics and ask you sort of where you are now in your work. And we talked about this earlier and the importance, building on all of this and where we are today, thinking about climate change, thinking about the population growth that we've got, the terrible crises that we're having with hunger and food insecurity. Is this and your work and this kind of thing the answer to the human dilemma and challenge to feed this planet?
I think not so much my work, but genetically modified organisms, which are also, you know, we talked about human genetic engineering, but plant genetic engineering is highly controversial in the public. And yet, you know, for scientists and in terms of the data available for any kind of harm, there is no demonstrated harm with billions and billions of meals of modified organisms. And really what genetically modifying crop plants do is speed up the breeding time tremendously. It both speeds up the breeding time and it allows the introduction of diversity that is not present within the species. And so with basically, you know, agriculture at all levels will not be able to meet the demands of humanity without the use of genetic modification. It's just not possible. We won't be able to derive the drought tolerant, pesticide resistant, climate, you know, all of the traits that are going to be needed to produce, you know, poor soil, interaction with microbes. All of these are going to require genetic modification of our crops. And it just really has to be. I think it's an amazing technology that is really coming along just when we need it.
We talked earlier about the controversy from way back in the 80s when our distinguished scientists here came forth with their work. And there's similar controversy around GMO, genetically modified organisms. Do you see that as a function of, I guess as the other case, a fundamental misunderstanding of what this is by the public? Or is there some real danger here, some real risk of playing God?
I mean, I honestly see no risk at all. I mean, there's certainly risks if you put something dangerous in, but there really is no risk in terms of improving a plant through the use of genetic modification. You know, playing God, I'm not going to address that. But you know, seriously, if we were running out of fresh water, our soils are depleted. I mean, the current agriculture is not sustainable in any way, shape, or form, even without extremes of climate becoming more dramatic, flooding, drought, I mean, heat, all of that. You know, the scientists are making tremendous progress to increase yields despite those extremes. And honestly, like I said, without it, we will never be able to meet the demands of not even growing populations, but existing populations on less and less land also.
I have a number of questions from the audience that I'd like to get to. One is a follow-up to this, so I direct it to you. And Allan and Jerry, if you want to jump in, please do. This is from Stephen, who asked, why does Europe have more strict food rules regarding genetically modified organisms?
It's a really good question. You know, I think part of it could be they had issues with mad cow disease. They've had real issues that have led them to sort of broadly ban things. But I think that, you know, I think it's changing in Europe.
I have a more cynical explanation for
Go
that.
for it, Alan.
That Chris Somerville, who was on one of the committees in Europe, told me, which is that most of the technology for genetic modification of plants was developed in the United States. And Europe was very afraid that all the best varieties and things, all the patented technology and the varieties would suddenly all be coming from the United States, and they just weren't going to accept that. So they decided to really get behind these attacks on the safety to keep the US from dominating this important technology.
Yeah, politics, nationalism, confusion, a lot of things. Karen asked this question, what are the functions of the transposable elements that make up half the human genome?
It's a really good question. The vast majority probably have no function at all. They amplify, and the organism, the insertions are neutral, they have no impact. But we're just slowly beginning to see more and more of their functions in terms of regulatory roles. I mean, Alan and Jerry were talking before about gene regulation, and you know, a lot of the diversity that exists in a species is due to regulatory changes, and many of those changes, or I don't know how many, but a lot of those changes are due to sequences introduced by transposable elements. So the vast majority, I would say, have no impact, no role. It's just very hard for an organism to get rid of DNA, large quantities of DNA.
I think they're more like, the genome is a separate ecosystem, and the transposons live in that ecosystem, and they have the ability to move and increase their numbers. But of course, they don't want to kill their host. So they are regulated in some way by the rest of the genome. But it's a complicated ecosystem. It's not that they're doing something for the human genome, they're doing something for themselves to propagate themselves within the human body.
But on top of that, the host can regulate these transposons by very sophisticated mechanisms related to RNAi. And you don't see any animals that don't have, or maybe all organisms that don't have transposons. And one of the reasons for that is very likely that periodically there's challenges, climate change being one example, but there's been one thing after another, new competitors, whatever. And it's quite likely that the animals that didn't have transposons didn't survive those challenges. So they might not be doing much for you now, but the next challenge, those organisms with transposons generate a lot more diversity than an animal without them. And so they have a much better chance to survive the challenges that are inherent in just the evolution of life.
So a follow-up from the audience, Nina asks, could transposable elements influence epigenomes?
Oh, absolutely. In fact, epigenetics probably arose as a byproduct of transposable elements.
And could you explain this just in case we have
Jerry was just talking before about the host being able to control transposable elements. The main mechanism that the host uses is to silence them, and it silences them using by a number of ways. One is to have the chromatin, intensely condensed into heterochromatin, which is an epigenetic phenomena. And it's what induces epigenetic regulation is insertions of DNA into places in the genome, which are different. And so that's why epigenetics, it's a mechanism that viruses are detected. So, so essentially, yes, it has everything to do with epigenetics. I'll say that.
Want to add anything, Alan, Jerry?
I think she covered it.
Okay, so to that point, Stephen asks, since you mentioned DNA, how much of junk DNA do you think is really junk?
Well, first, I don't think anybody working with this likes it to be called junk. And how much is junk? It's unknown. I mean, I'd say at least half or 3/4 of it, if not more. I mean, the stuff that's condensed into heterochromatin that really has sort of silenced. So it's a large percentage of what's there, I guess, could be called junk, but we don't know. We don't know.
Allan, I want to change the subject slightly for a couple of minutes, and I'll come back to some other audience questions about the Drosophila, the fruit fly. That makes it, and what is it that makes it so genetically connected? And you're working on a variety of things to include trying to better understand, for example, muscular dystrophy in human beings. Could you talk about this?
Well, I don't know if I haven't done very much on muscular dystrophy, but I have done a lot on the ovary and the process of female reproduction. And some organisms go on an evolutionary, path off by themselves, but then others stay in a kind of mainstream that all the other organisms I followed, and then, before they branch off somewhere. So what we found, I first spent a lot of time, in fact, I really wanted to develop transformation so I could continue my work understanding the ovary and how it worked in the fruit fly, which is what I did my postdoc on. And we made it over time, when you could now, with this new technology, do genetics much better. We made a lot of progress, and many other people contributed huge insights. And then, 20 years ago, I started saying, I wonder, if we could do the same thing in the mammal. And I started working on mice, and sure enough, the female reproduction in the mouse is just coming to look so similar, the same order, the same genes, the same kind of cells. So it's very, very relevant to use model organisms to get a speeded up, and we can do experiments so much faster in Drosophila, and then go to the mouse and, oh yeah, look, it's the same. So it's just a very, very efficient, it's a very powerful way to work. It's not something that just crazy professors want to do. This is like the smartest thing to do if you want to get the answer the fastest and the most accurate.
And this is where your lab is working, and the basic knowledge here has a lot of implications, right? For, as you say, fertility and infertility, in vitro fertilization. Does it extend to birth defects and understanding some of these sorts of things?
Well, birth defects are very tied up to reproduction. And just consider this question. We don't know how similar each egg is in an ovary. And if there's some differences, some of those differences might increase the chance of a defective developmental process. So until you actually can go in and look at questions like that and determine why would there be differences, what sort of differences, and do they in fact, or which ones of the differences are important for birth defects, how will we ever understand why we have this problem of birth defects? I think you just have to start going through the biology until you really understand it, and then we'll know a lot more about how to minimize birth defects.
I have another question from the audience I want to go to, and Jerry, I'll let this one come to you. This one's from Eric, and he says, given the previous challenges with transformation, can you reminisce further if you had to solve particular issues to get the first transformations to work? Did you work with anything besides P transposon? Any general lessons about how to get something to work when it's never worked before?
Well, we had thought about, transposons as vectors before, but it was when we found out the unique properties of the P element that they would transpose at much higher rates than other elements under conditions which we could control. It really made it clear that this was the one, to try. So I think that if you want to do difficult things in science, it's good to be in a supportive environment where you won't lose your job if you fail, which Allan and I were lucky at Carnegie. It's good to have good collaborators who you can bounce ideas off of and develop and perfect your ideas and figure out what we should try and what wouldn't try. So we did this for hours a day, we'd be talking when we're in the code room together. Well, what's the right way to do this? What if this doesn't work? How should we do it next time? And it's a very iterative, I think, process, and it's fun. The main thing to say, it's fun. It's fun. It's challenging. It's you against Mother Nature. It is a real enjoyable experience. Or if it isn't for you, you shouldn't be a scientist, I guess. That's what makes scientists fun. But I could respond a little bit to this other argument about flies. So right now I work on the fruit fly brain and how fruit flies learn and remember and process information. I work on aggression, between flies, all these things that you wouldn't think that flies do all these things. And I think there's a strong belief among biologists in evolution that once some mechanism of doing something has evolved, it will be enhanced and modified, but the core things will be the same. So if you look at a fruit fly neuron and at the various molecules in it that involve in signaling from one cell to another, and other things like that, these were all ancient, before the last common ancestor of a fruit fly and a human diverged. So I think we can learn stuff about how brains process information and what makes the way biological computers, if you will, different than kind of silicon computers. So these general rules, you can almost study anything, and it's very exciting to look at these.
What are you learning, and what are you learning about aggression and what we see in fruit fly brains, and God forbid how it might apply to human brains? And
well,
what
it's the same molecules. When you do something and you get a satisfaction, there's a release of dopamine in your brain. It's a positive thing. It's the same true for the fruit fly. It's the same molecule, it's dopamine being released that gives you a reward signal. It's the same logic that's used by a fly to learn something. It tries it, and if it doesn't work, it doesn't get the dopamine. If it does work, it gets the dopamine, so it reinforces that behavior. So a lot of these things are, the molecular mechanisms seem to have been discovered early in evolution and seem to be being maintained. So I think, we're not going to understand human consciousness by studying the fly, but we'll understand a lot of things about how organisms do things and can accommodate things, can go to a particular point in space, can navigate. Flies are pretty good. If you try to swat a fly, they're pretty effective at avoiding you, and they can move around the room at high speeds without crashing into the walls. They'd be much better than any self-driving car can do. So,
another question from the audience here, and I think this will bring us back to Carnegie and Don Brown and some of these other things as we prepare to wrap this conversation. This is from Martin, and he's asking, does the panel think there's more pressure on patenting discoveries now versus in the 70s and 80s?
Well, I think I don't think we knew about patenting much. And certainly when we made our discovery, Don Brown said we should patent this because in the past, Carnegie had made many discoveries that if they had patented them, they would have led to a source of revenue. So we should patent P elements. So Allan and I wrote a patent, and we had lawyers, and we did patent P element. And it didn't end up paying off for Carnegie in some sense. It did get licensed and did give them some royalty. But that was my first experience.
Well, I would just point out that compared to most academic patents, the P element patent, by a crazy process, turned out to be pretty profitable for Carnegie. I mean, the total revenues to Carnegie from the P element patent were somewhere between $2.5 and $3 million. And, that's not that much on the scale of industry, but it's not exactly nothing either. That farm been paid all the work, probably paid all of our salaries, whatever. So it's just the process of patenting, in my experience with this, is it just shows how flawed the process is. It's not even as good as the process of allocating scientific credit, which has its own problems. But the patent system of deciding what's true or not, I don't know. I just will recall we tried to make another patent, and the examiner told me that no, that's not novel because this paper already showed that. And I looked at his paper. I said, but wait a minute, that's my paper. I wrote that paper, and it didn't show that. And he said, no, I read it, and it shows that. It's a crazy system. That does not really get at the true scientific concept of truth very often.
Okay, but on the positive side, certain things need to be patented to bring them to development. So if you're going to make a drug that people are going to take as a pill, that needs patent protection because of the billions of dollars that have to be invested by a company. So we're not saying, I don't want to do this as patents are bad, but I would say that most universities or institutions like Carnegie in general don't make money on patents.
Sue, what's your perspective on this?
I think that since Carnegie, and I mean, since that time, not so much since Carnegie, universities have in their research office, there's a real focus in patenting. And I think that they overemphasize it greatly. I think that, we're quick to put in a disclosure notice. And I think it really, I think universities think that a lot of the stuff we're doing is a lot more valuable than it really is. I think the value really comes in the basic research that it enables. But yeah, I think for most researchers, there is a pressure from the vice chancellor, vice president of research office. And I think that it's way out of line with, the real value that could come to the university.
Jerry, I have a question that's directed at you from the audience, from DJ, who says, did you and Allan already conceive of the idea of using P-element to transform flies before moving to Carnegie? Did you move from Harvard to Carnegie to specifically work on the transgenic fly project?
So when I moved from Harvard to Carnegie, I hadn't yet cloned P-elements. So my first project, I was working on transposons, we didn't yet have the P-element. So that happened early in my time at Carnegie. And once we had the P-element, Allan and I realized it would be the ideal vector, ideal transposable element to try this with. The idea that I had in my mind that transposable elements might be good for doing gene transfer, but the transposable elements I was working on before the P elements wouldn't have worked for this. So it was really, it developed at Carnegie, and the impact and the willingness. And again, I wouldn't have taken that risk by myself. It would have been, it was having Allan around and say, yeah, let's try this. Yeah, I'll get you, don't worry about injecting the embryos. I'll figure out how to do that. You just make the DNA construct. It built on itself. It was a unique constellation of people and ideas. And so
I would say the more we talked about it also, which we talked extensively before we ever started doing experiments, it became clear that there was a lot of reasons to think this would succeed. The P elements had come to Drosophila melanogaster from a distant species. And then they had spread throughout all the world of Drosophila in a very short time, like 20 to 25 years. So that sounds like an element that can if it gets in, it can jump and it can be effective. It's very different than your run-of-the-mill transposon that's been in this genome already for millions of years and it's not doing all that much, but maybe every once in a while it jumps somewhere. So we realized that not all transposons are equivalent. And the trick is to get the right transposon for any given species. And we believe that our method would work in any species if you just had the right element to choose from, one that wasn't already there. And then once we did transformation, it was validated this. Like if we injected P elements into a strain that already had P elements, you got 0 transformants. If you injected it into a species strain that didn't have, that didn't have P elements, you got lots of transformants. So it was the absence of being there to start with, because there's repression develops, and if that hasn't, it has to have not developed to be successful.
So here's a perfect follow-up question from Jiang Wang, who writes, what were the specific key technical challenges and tricks that led to your success with P-element transformation?
Well, there weren't any genes there were no genes that were known to be on a piece of DNA. So how can you rescue something? You need a mutant and you need the corresponding piece of DNA, but there was no way to know if it was right. So that was one idea, but Jerry came up with a very clever assay, in terms of using an unstable mutant allele to tell if a P element had gone into the genome, because now that this allele would stay unstable, and that worked perfectly. It took cleverness and thought in the environment of that time when there were so few molecular tools,
and a lot of luck, I would say,
and a lot of
luck. I mean,
the piece that we took, which we didn't even know had the Rosie gene, it was like 7 kb, and the Rosie gene turned out to be 5 kb. So if it hadn't been precisely right in the middle of that 7 kb, it just wouldn't have worked.
Robert's kind of curious about how this happened. Question from the audience. Did Allan take part in recruiting Jerry to Carnegie? At the time, Robert writes, Harvard had a stronger reputation than Carnegie. So we're getting down to it.
I could tell this story. It won't make Harvard look very good, but that's not my problem. I was at Carnegie and at Harvard, and I at a cancer center at Harvard Medical School. And Allan was on the job market. He was a year later or so than me going on the job market. And he interviewed at Harvard. And then we had a faculty meeting to decide whether to offer him a job. And my immediate boss said, Allan is a great scientist. He seems like a fine young man, but no one who works on Drosophila would have any long-term future in a cancer center. So this did two things. It made Allan didn't get the job, and I learned that I should be looking around to move somewhere else. So after Allan got hired at Carnegie and there was an opening there, he wrote to me and said, are you interested in perhaps moving to a more supportive environment? And that's how it really started. So Harvard was it was more well known. I don't know if it was more prestigious among scientists, but it was more, well known. Certainly my mother had heard of it, and she probably hadn't heard of Carnegie. But I think as encouraging as a place to be and to feel like you really were a scientist, you were supported to be a scientist, it was a really unique institution. So
well, and this is part of Don's legacy and part of Carnegie, right? Taking the risks, bringing the scientists in, letting you do your thing, support it. Last question, I said a moment ago, but this one I want to get to from the audience, and this is to all of you, any of you, and it's a great broad question. Do we know more about animal genes or plant genes? What information gaps are potentially the most transformative? Who wants to go for that one? Sue?
I think we probably know more about animal genes because a hell of a lot more money is spent on animal biology than plant biology. And I think to move things more quickly in plants, we need more money. I mean, it's as simple as that. There's a huge underinvestment in plant research in this country. Quite honestly, to hear our president, who I otherwise respect, talk about another moonshot for cancer was really depressing, considering because the impact would be on a lot of older, rich people. Whereas for plant science, agriculture is the largest ecosystem on this planet. And as I said before, we are facing a grim future if we can't understand the basic biology and just everything about plant sciences. We fund less since 2006. China, many countries have funded more in plant science than our country. We don't have the researchers. We don't have people to go into positions in industry. We're not training enough people. Everybody's interested in biomedical science.
You are three remarkable scientists who have done such incredible things, as we've discussed here over the last several decades. And you're getting to an issue now, Sue, about funding and priorities. The other thing that's happening out there is, in many places, science is under siege. It is being attacked. It is in the public sphere. We've seen it with COVID. We see it with climate change. We see it with GMO. We see it with all these things. What do you think your colleagues, scientists, what do you think others in our culture can do to help science explain and defend itself, which appears to be increasingly necessary in this very toxic environment we're living in now? I realize I'm putting you all in a different place than we've been discussing here, but you've got such experience and such insight. I think it'd be very interesting to hear your thoughts on this. Anybody want to volunteer to go first?
Well, I'll just say, I think we live in a different environment now, where there isn't a universal acceptance of what is true and what is not true, and how you determine what is true and what is not true. So in the news media, when I was growing up, you listened to Walter Cronkite, and he would tell you stuff and you would believe it. And it wasn't like an opposing set of views. It wasn't like he said black and there were other people saying white and you were left confused. So I think there isn't just a general level of trust and a general level of how you determine what is true and how you know whether you can believe something or not. I think it's sort of, in that environment, it's sort of depressing to know how to make progress, because you don't have people with an open mind who you can really discuss your views with. Your views may be wrong and they may be right, but there can't be a kind of debate with well-meaning individuals to try to reach a consensus. There doesn't seem to be any forum for that anymore.
Alan?
Well, I think that one reason that this has happened, that hasn't been true much of the time in the past, is that we've had a very, very prosperous period, and people don't see a connection between truth and whether they're risks, that they're taking, or whether they, have the necessities of life. But of course, that can change. And, if we go through, either due to climate change or some other unfortunate events, a period of much greater marginality to human life, then the truth will matter again.
So,
the real truth.
Well, the science, yeah.
Yeah, go ahead.
No.
I think that, in addition to underfunding plant biology, we're also underfunding the social sciences. And whereas we can incredibly quickly develop a vaccine, our science has brought us to the point where we can develop an incredible series of vaccines in record time, we still couldn't figure out how to convince people that these were good for us. And I think the same thing is happening, with GMOs, with climate change. I mean, every area. And we really need to, beef up funding in the social sciences, because like other areas, it's really, really underfunded.
Let's conclude with this. I mean, we started by talking about Don and his impact and the influence he's had on each of you and how you worked in this lab. And here we are talking about science. And sort of as you think about the future of science, what are the lessons you think that Carnegie and Don Brown's leadership and management of this remarkable research, what should that message be to young scientists and others going forward?
I mean, I think what was so omnipresent at Carnegie was the enjoyment that all of us had with science. And I think a lot of that has been lost now. I think funding is much more competitive. It's not nearly as much fun as it used to be for young people to do science. I think it is for a subset, but there's just tremendous competition. There are few jobs in certain areas, and there's tremendous competition for funding. It's that comes first, and it's very sad to say it's not the same business that it was when I.
Anything done about that, Sue?
I don't know. I
Hmm.
don't, I mean, money helps, you know?
Yeah, yeah. Jerry?
I agree with Sue that science, I had the impression that science was more fun for us in those days at Carnegie than it is for the people I see now doing, young people I knew doing science now at current institutions. And I think they have a lot more stress on them. I think people's careers got delayed. You know, we were very young, you know, we were already, when we did our best work, because we got to be independent scientists early on. And the average scientist the age that Allan and I were when we did this work is still an apprentice in someone else's lab. They haven't yet gotten the ability to be independent. So I think all these things are not helpful. We trained a lot of scientists, but we didn't provide funding for them. So everything is more competitive. And so I think it's not as easy now as it was in the good old days for us. And I don't think any one thing is gonna cure this problem.
Allan, can you leave us with an inspiring thought?
Well, I think the thing that hasn't changed is what leads to success in science. And, you know, the people who will continue to be really successful will have to find a way, like every scientist in every era has done, to get enough funding to be able to follow their dream, to find some little niche where someone isn't paying that much attention to what they're doing long enough for them to do the experiment they dream about, and then to actually accomplish something that really makes a big difference in science. And I think that is going to continue to happen. It's really sad to think we can't make it more widespread and efficient, and that we might even be going in the wrong direction. But the rules of science for success really haven't changed, and Don Brown seemed to know an awful lot of them.
Well, Don Brown has been with us today, so I want to thank you, Don, again for the incredible impact and influence that you have had in your science and your life and inspiring other scientists. I'd like to thank the three of you, Sue and Allan and Jerry, for your time here today and your insight and all your work, and our colleagues at Carnegie for making these conversations and symposia possible for us to reflect on both what Carnegie has contributed and on the work that each of you has done. I want to thank our audience for some really terrific questions for the conversation today, and tell you to kind of stay tuned, because they're working on some more of these conversations that will continue to honor Don and his work as he celebrates his 90th year, and Carnegie as it continues to celebrate and pursue its very, very important work. So, folks, thank you all very much. It was a great conversation. Sue, Allan, Jerry, really appreciate it.
Enjoyed it.
Thank you, Frank.
All right. Good luck to everybody, and thank you so much for joining us. I hope we'll see you again soon.
Take care. Thanks.
We hope so too.